When we think about the history of life on Earth, we often imagine a gradual progression. Simple organisms giving rise to more complex ones, primitive bodies slowly transforming into familiar forms, fish becoming amphibians, synapsid giving rise to mammals, dinosaurs evolving into birds. This narrative, although broadly correct, can give the impression that evolution follows a predictable path, that over time, life refines itself toward recognizable and stable designs. However, if we travel far enough into the past, long before forests covered the continents, and long before vertebrates ever set foot on land, we encounter a world that challenges this perception entirely. Let us travel back more than 500 million years ago to the Cambrian period. During this time, life was almost exclusively marine. The continents were largely barren landscapes of rock and sand. Yet beneath the surface of shallow seas, evolution was undergoing one of the most remarkable radiations in the history of our planet, the Cambrian Explosion. Within a relatively short geological interval, most major animal body plans appear in the fossil record. Organisms evolved hard parts, complex sensory organs, new feeding strategies, and unprecedented ecological interactions. Predation became widespread. Mobility increased. Marine ecosystems grew more structured and dynamic. Among the extraordinary creatures that inhabited these early seas was an animal whose anatomy initially puzzled even experienced paleontologists. Its name is Opabinia regalis. The fossils of Opabinia were discovered in the early 20th century in the Burgess Shale of British Columbia, one of the most significant fossil deposits known to science. The Burgess Shale preserves soft-bodied organisms in exceptional detail. It offers a rare and invaluable glimpse into early marine ecosystems that would otherwise be almost entirely unknown. When the British paleontologist Harry B. Whittington redescribed Opabinia in the 1970s, his careful anatomical reconstruction revealed a combination of features unlike those of any modern marine animal. Opabinia was a relatively small organism, measuring only a few centimeters in length. It possessed 5 stalked eyes positioned dorsally on the head, a configuration that appears highly unusual among early arthropods. It has been suggested that this arrangement may have provided a wide field of vision, potentially advantageous in detecting prey or avoiding predators in the complex seafloor environment of that time. Extending from the anterior region of the head was a long and flexible proboscis, terminating in a grasping structure equipped with small spines. This appendage may have functioned as a feeding organ, allowing the animal to probe the sediment and capture soft-bodied organisms. Fossil evidence indicates that Opabinia possessed a ventrally located mouth, oriented posteriorly beneath the head, a configuration that initially generated considerable debate regarding its functional interpretation. Along the sides of its segmented trunk were lateral lobes, which likely played a role in locomotion. These flaps may have moved in coordinated undulatory waves, somewhat comparable to the swimming mechanisms observed in certain modern marine invertebrates. Beneath these lobes, delicate gill-like structures are thought to have facilitated respiration. Despite its highly unusual appearance, Opabinia was not an isolated evolutionary anomaly. It is generally regarded as part of the arthropod stem lineage, a form closely related to true arthropods, but outside the group that includes their modern representatives. Today, Opabinia is understood to represent an early branch of the euarthropod lineage, diverging before the emergence of true modern arthropods. This interpretation is supported by the presence of a segmented body plan and by structural characteristics of its appendages that suggest affinities with early euarthropods. Nevertheless, its precise phylogenetic placement within this early radiation remains the subject of ongoing research. Current phylogenetic evidence indicates that Opabinia and radiodonts such as Anomalocaris belong to the stem group of euarthropods, early evolutionary branches that diverged before the emergence of true arthropods. Although they represent distinct lineages within that stem radiation, their close evolutionary relationship is well supported. In earlier classifications, these Cambrian forms were sometimes grouped together under the name Dinocaridida, reflecting their shared position near the base of arthropod evolution. The description of a second Opabinide, Utorora camosa, from Cambrian deposits in what is now Utah, suggests that this lineage may have been more diverse and geographically widespread than previously recognized. What makes Opabinia particularly significant is not only its distinctive morphology, but also what it reveals about the broader evolutionary context of early animal diversification. At that time, the range of anatomical diversity appears to have been broader than in later intervals, before many lineages were eliminated through extinction or evolutionary replacement. Opabinia may represent one of the many early lineages that arose during this interval of rapid diversification and appears not to have left surviving descendants beyond that time. The Burgess Shale has yielded fewer than 24 well-preserved specimens of Opabinia. From these rare fossils, paleontologists have reconstructed its anatomy with remarkable precision. Nevertheless, several aspects of its biology remain uncertain. It is not entirely clear how rapidly it was capable of swimming, how it reproduced, or the exact ecological niche it occupied within these early marine communities. It is generally thought that Opabinia functioned as a small predator or scavenger, feeding on soft-bodied organisms living on or within the sediment. Its flexible proboscis may have been particularly suited for extracting prey from crevices or shallow burrows. However, direct evidence of gut contents is limited, and certain aspects of its feeding strategy remain hypothetical. By the middle of the Cambrian, animals such as Opabinia coexisted with an increasingly diverse assemblage of trilobites, sponges, early mollusks, radiodont predators, and numerous other enigmatic organisms. Marine ecosystems were becoming progressively more complex, and ecological interactions more intense. Over time, lineages more closely resembling modern arthropods became dominant. The distinctive body plan of Opabinia did not persist beyond that geological interval. Whether this disappearance resulted primarily from ecological competition, environmental changes, or broader evolutionary contingencies remains difficult to determine. What is clear is that Opabinia occupies a crucial position in our understanding of early animal evolution. It serves as a reminder that the history of life is not a simple, linear progression toward modern forms. Rather, it is a branching and dynamic process shaped by diversification, extinction, and survival. If we observe the animals that inhabit our oceans today, their body plans appear coherent and stable. More than 500 million years ago, such stability had not yet been established. Opabinia stands as a reminder of that early phase of biological innovation, when animal body plans had not yet stabilized into the forms that would later dominate marine ecosystems. Although it vanished hundreds of millions of years ago and left no confirmed descendants, Its fossils continue to inform our understanding of how complex life diversified so rapidly during this foundational chapter of Earth's history.